All right, today we're going to take a journey into the life of what is, without a doubt, the most successful life form on our planet. And nope, it's not what you're thinking. We are demystifying the prokaryote.
So, let me just start with a question for you. When you think about the most dominant, successful life on Earth, what pops into your head? Is it us, humans? Maybe insects? Or hey, maybe even plants, right? Well, the answer is actually none of the above. It's this little guy, the prokaryotes. I mean, these single-celled organisms have been around for billions of years, and they outnumber every other living thing on this planet, combined. They are the true masters of survival. To really understand them, you gotta go way back to the very beginning. Prokaryotes are essentially the original blueprint for life. They're the foundational design that all other, more complex life, yeah, including us, eventually came from. Okay, before we get any deeper, let's make sure we're all on the same page. What's a cell? It's the basic building block of life. The best way to think of it is like a tiny, self-contained factory that has everything it needs to function. All life is built from these. Simple as that. You know, when you put a simple prokaryote next to one of our own complex cells, a eukaryote, the differences are just wild. For starters, prokaryotes are way, way smaller. Their DNA is just a single circular loop floating around inside. They don't have a nucleus or any of those other little membrane-bound organs or organelles that our cells are packed with. They are truly the minimalists of the biological world. So let's start our tour of the prokaryote from the outside in. We're going to explore this incredible three-layer defense system that protects these tiny cells from a, let's face it, pretty hostile world. On the outside, prokaryotes have some amazing gear. A lot of them have a flagellum, which is basically a motorized propeller that lets them swim around and find food. Then you've got the capsule, which is the sticky, sugary outer shield. Underneath that, there's the super important cell wall, which gives the cell its shape and strength. And finally, you hit the plasma membrane, the bouncer at the door that decides who gets in and who gets out. Now, that cell wall is especially important. Its specific makeup is actually the main feature scientists use to classify bacteria, with a technique called the Gram stain. Basically, the kind of armor a bacterium is wearing tells us exactly who we're dealing with and how to fight it.
This Gram stain test splits bacteria into two big teams. You've got gram-positive bacteria, which have a really thick outer wall made of something called peptidoglycan. Think of it like a thick brick fortress. Then you have gram-negative bacteria. They've only got a thin layer of that brick, but it's covered by a whole extra outer membrane that can be really toxic to us, which makes these guys particularly dangerous.
So what exactly is this peptidoglycan stuff? Well, imagine it's like biological chainmail. It's the super strong mesh-like material made of sugars and proteins that gives the cell wall all its strength. And here's the kicker. This very structure is the bullseye for some of our most powerful antibiotics, like penicillin, which works by literally stopping the bacteria from building this mesh armor. Okay, we've made it past the armor. Let's go inside the cell. Now, if prokaryotes are minimalists, their internal design is where that philosophy really shines. So, what's it like in there? It is life with just the bare essentials. There's no nucleus, no powerhouses like mitochondria, none of the complex compartments you'd find in one of our cells. Everything, the DNA, the little protein-making machines called ribosomes, all of it is just floating around together in this thick internal soup called the cytosol. It's maximum efficiency, minimum clutter. Now for the most critical part of any cell, the instruction manual. Let's unpack the prokaryote's genetic secrets, including its main chromosome and what I like to call its shareable upgrade packs. So prokaryotic DNA comes in two flavors. First, there's the main bacterial chromosome. You can think of this as the cell's core operating system, a single circular loop of DNA with all the essential code. But then you have plasmids. These are tiny extra circles of DNA that are kind of like software plugins or upgrade files. They carry non-essential genes that can give the bacteria cool new features. And why are these upgrade packs so incredibly important? Well, this is where it gets serious for us. The ability for bacteria to trade these plasmids back and forth is one of the biggest reasons why antibiotic resistance can spread so terrifyingly fast. It's a massive challenge for modern medicine. Now, these plasmid circles can physically exist in a few different shapes. The easiest way to picture it is to think of a rubber band. In its most perfect stable form, it's what we call a covalently closed circle, or CCC. both strands of the DNA are totally intact. But what if you take that rubber band and just snip one of the two strands? Well, all the tension gets released. The plasmid relaxes into this floppy, less compact shape. We call this the open circle, or OC form. And finally, to save space inside the cell, that original tight rubber band can be twisted up on itself into a super compact little knot. This is the supercoiled form. And believe it or not, understanding these different shapes is the key to how scientists actually work with them.
Speaking of which, scientists have gotten really, really good at, well, borrowing these plasmids for our own purposes. So how do we actually pull off this microscopic heist? Here's a quick 5-step guide to how it's done.
The whole process is really just a clever chemical trick. First, you grow billions of bacteria, and then use chemicals to burst them all open. Then comes the magic step. You add a solution that messes up all the DNA, but only the small, tough little plasmid circles are able to snap back into their original shape. The huge bacterial chromosome is left behind as a tangled mess. After that, it's just a matter of cleaning up the proteins and pulling out the pure plasmid DNA.
So we've journeyed from the outer armor to the inner genetic code. Let's zoom back out for a second. Why does all this matter? Why does understanding these tiny, simple organisms have such a gigantic impact on our world? It really comes down to tiny, fundamental differences, which can be summed up by numbers like this one, 70S. That's the size of a prokaryote's ribosome, its protein factory. Our ribosomes are bigger, they're 80S. It seems like a tiny detail, right? But that one difference is exactly what lets some of our best antibiotics kill bacteria while leaving our own cells completely unharmed. We literally exploit their minimalism to our advantage. And that brings us to the ultimate two-sided coin of the prokaryote world. On one hand, we've learned how to hijack their plasmids, turning them into amazing tools for cloning genes and producing life-saving drugs. But at the exact same time, they use those very same plasmids to pass around antibiotic resistance genes, fighting back against us. It is a full-on, high-stakes biological arms race. And that leaves us with one final, incredibly important question. These simple cells have been adapting and surviving on this planet for billions of years. As they continue to evolve brand new ways to resist our best medicines and technologies, the real challenge is, how will we evolve with them?